Great. Thank you so much. I appreciate the opportunity to share with you some of our recent findings and our work. And today I'm going to tell you about our discovery that robust B-cell responses predict clearance of Borrelia infection and return to health. So I'm going to start by discussing use of antigen microarrays to profile antibodies in response to Borrelia infection and their associations with clearance versus non-effective control of that infection. I'm then going to describe how we applied a sequencing and antibody repertoire sequencing technology developed in my lab to sequence antibody repertoires in acute Borrelia-infected humans, and then use of those repertoires and sequence data sets to identify Borrelia-killing antibodies that we think could provide the basis for next-generation therapeutics. So first, there's a long history of investigation of the role of antibodies against Borrelia burgdorferi. It's well known that there's a lot of heterogeneity in patient antibody responses, and this is obviously a major challenge in developing effective diagnostics. And as you all are very familiar, there's a current diagnostic paradigm where it's called the two-tier testing, where you first have to have a positive ELISA test, and then they follow that with a screen on a Western blot, and you have to have a certain number of bands to be called, positive. And this, it suffers from limitations in both its sensitivity and its specificity, and it's often misinterpreted. And another kind of important point is that these antibodies are not only diagnostic in utility. If you transfer antibodies to mice, for example, from people who cleared the infection, they can protect the mice against the infection. And further, there's their role in potential autoimmune complications in a subset of people who develop post-treatment Lyme, is not that well defined. And I'm going to show you evidence that some of these antibodies generated, in fact, target host proteins, in addition to other ones targeting Borrelia. So many of our studies are in collaboration with John Acott at Hopkins, who's been mentioned multiple times and will be involved in a panel discussion later, moderating a panel. And we're using and leveraging his cohort, the Study of Lyme Immunology and Clinical Endpoints, or SLICE. This is a 2-year prospective cohort. At the top are the Lyme cases, so people who actually had Borrelia burgdorferi infection. And the bottom is the controls. You can see over a 2-year period, there's multiple samples collected, and we're characterizing these samples as they relate to the clinical outcomes. So clearance of the infection and what we term return to health, or development of complications, which is termed post-treatment Lyme disease syndrome. And so basically, including these slice samples, we analyzed on a antigen array assay that we developed samples from over 5,000 patients from multiple different sources that are listed here. And I'm going to just highlight a couple punchlines from our discoveries. The first is that individuals who are infected with Borrelia burgdorferi who mount robust antibody responses against certain Borrelia proteins. So out of kind of several hundred Borrelia proteins and peptide antigens on our antigen arrays, this small set of about 15 Borrelia antigens were the ones targeted by antibody responses in individuals that return to health, which is on the right side as compared to people who develop post-treatment Lyme disease syndrome. These antibodies target Borrelia antigens, including VLSE, outer surface protein C, BMPA, decorum binding protein B, and importantly, and I think which reinforces this, is you'll see in our repertoire sequencing data sets, kind of 10 slides from now, these are the same targets that the activated B cells are making antibodies against. So we're seeing it in the pooled serum. These antibodies correlate with return to health, and we'll see it subsequently in the B cell repertoire sequencing data sets, and then we're further converting these antibodies and testing them in collaboration with Monica Embers and Mickey Tall to demonstrate which ones actually have killing activity. We also profiled cytokines in these, and we identified increased levels of IL-33, a key cytokine or little immune signaling molecule that is associated with return to normal physical function, as indicated here, as opposed to people became, in the slice cohort who became disabled. And we think this might be an important master cytokine because it has both transcription factors, meaning it's altering the gene expression in the cell, as well as acting as a cytokine. And it drives T cell responses that are effective at clearing, parasitic infections, including spirochete infections such as Borrelia infection. So just to summarize our profiling of antibodies and cytokines in Lyme, we've published several papers on this. We developed this Borrelia burgdorferi antigen array. We basically started with a large planar array, got down to 62 candidate antigens, and then basically refined that down to a 10-plex panel. And recently, this panel, based on collaborative work with Bio-Rat, has been 510(k) cleared by the US FDA as a next-generation serologic test for Lyme disease. This was in April 2019, so just a few months ago. And this serologic assay is meant to be more robust than the current two-tier testing paradigm. It provides about a 57% sensitivity at presentation and an almost 84% sensitivity post-treatment, and it's 98% specific. So we hope that this panel in this now 510(k) cleared test, which is a higher bar for approval than a CLIA lab assay, will advance the care for patients. And second, we identified this master transcriptional cytokine regulator IL-33 as potentially playing a role in driving responses that control infection. So just summarizing this preclinical development on the left side, that was in my lab in collaboration with Bio-Rad, and then translating this finding, working with Bio-Rad into this 510(k) cleared test that's available in many labs throughout the US and the world. So I'm now going to shift and talk about some of our repertoire sequencing work. We developed technologies to profile antibodies, including more recently cell barcoding technologies that we use to sequence antibody repertoires. We take blood from patients, such as people with acute Borrelia infection or other autoimmune conditions or with cancer. We isolate certain B cell populations of interest, for example, the activated B cell population or plasmablast. We use barcodes. We then do next generation sequencing. We identify antibody repertoires. And just to summarize what we're doing, so first of all, we're applying this approach to blood plasmablasts. So these are the activated B cells of the moment in an individual. These are the ones being released from the lymph nodes in an ongoing immune response as being kind of the chosen B cells. So they're the ones that an individual human is producing at an individual point in time. And very powerfully, they're released into the blood, so you can do a blood draw and capture these B cells, and by sequencing them, identify the antibodies they're producing, and thus the targets of the current ongoing immune response. And if you do that in patients, for example, who clear their Borrelia versus develop post-treatment Lyme syndrome, perhaps we can gain insight into the protective and pathogenic responses mediating both. So that's kind of the overarching strategy. And this slide kind of summarizes how this approach works. If at the top level there's the naive B cells, and certain B cells become activated against, for example, in this case, Borrelia infection, and they produce daughter B cells. When we sequence repertoires, we present the repertoire at a single point in time as these evolutionary or phylogenetic trees, the tree displayed at the bottom. And you can see, for example, on the left side, the green daughter B cells are represented in the black circle green clonal family. And the blue daughter B cells are represented by the blue clonal family. And so this is basically taken somebody's active B cell repertoire and digitally distilled it down into the individual B cells that are generating those antibodies. And then we do something that I think is equally kind of unprecedented, not per se now in 2019, but historically in biology, is that we do what is effectively synthetic biology. We take the centermost clone, We take the consensus sequence, we email it to a gene synthesis company, and they just gene synthesize the DNA encoding the antibody, and then express it, and by FedEx, a month later, the antibody arrives back, and then we can characterize its functional properties. And this is, people may not appreciate this, but like certain people's PhD theses back kind of 15 years ago were solely focused on making like a single antibody and characterizing it, whereas we're able to now do this for many tens or hundreds of antibodies in a high throughput fashion, which is powerful to gaining insight on the mechanisms of disease. So this is our work showing first that in acute Borrelia infection, there's elevated blood plasmablasts, these activated B cells in the blood. Normally, they're present at very low levels. And so you can see just on the left side, we use flow cytometry to identify these. This is just the gating scheme by how we identify them. And then on the right side, plots their levels. And you can see that basically at baseline, when people present with erythema migrans, so rash, with acute Borrelia infection, there's like a 20 to 30-fold increase in levels of these activated B cells in the blood, and it tapers down over time. You can see at 6 months, it's basically back down to baseline where the line is across. But importantly, in the untreated population, you can see there's skewing. There's some people with high blood plasmablast, activated B cells, and there's others with low. And you'll see it's these people with the high levels that clear and basically return to health, and the people with the low levels who develop post-treatment Lyme. So this is another observation that we made from our sequencing data, and basically that in acute and chronic Lyme, there's skewing towards an IgA response. So this is IgA B cells come generally from mucosally activated triggers. So the left side is IgM. There's no difference from Lyme, which is the first 3 bars, to healthy. And the second middle is the IgG plasmablast, which is kind of the established ongoing response, where you can see the first 3 that are Lyme, early, late, post-treatment Lyme, are all low on IgG, whereas the healthy on the middle right bar is high. And on the right side, the Lyme, so both early Lyme disease, late Lyme disease, and post-treatment Lyme are all high for IgA plasmablasts in the rightmost graph as compared to healthy, which are low for these B cells. So it suggests that exposure at surfaces in the body where IgA B cells are activated are prominent in all stages of Lyme disease. We further show that in people with disseminated early infection, which means that John Adcock characterized them as having multiple erythema migrans, so multiple sites of rash, or disseminated rash, these individuals had a higher initial activated B cell response as compared to people who only had a single rash, localized. And this may more potently activate the immune response. And then here you can see these individuals at baseline, so they're untreated when they present with rash, who had high activated B cells in their blood, plasmablast, return to health overall as compared to people who at baseline presentation had low activated B cells. But that by 1 month, it's kind of a wash. Everybody's kind of converging. But at this point on John's protocol, they're all receiving treatment, doxycycline or another appropriate treatment for their infections. So we were very intrigued by this observation that high activated B cells was associated with clearance and return to health. And we sequenced their repertoires using the technology. So based on our sequencing data, first, we found that in individuals, so these are cord diagrams tracking across the slice patient visits over the 2-year period, persistence of activated B cell clones. And you can see at the bottom, there's graphs or cord diagrams for individual patients. And in general, the B cell clones don't persist. They mount this active B cell response, and the clones disappear from the blood pretty rapidly, probably due to clearance of the antigen. Whereas on the top are people who develop post-treatment Lyme disease syndrome. And you can see in these individuals over the SLICE study, so a 1 to 2 year period, there's persistent reactivation of multiple plasmablast or activated B cell clones, suggesting that there's persistent of the antigen stimulus to which these B cells are reactive to, and thus making them persist over time. So I'm now just going to show a couple repertoires, and then we're going to start looking at what the antibodies that are encoded actually target. And so this is just showing an example of a antibody repertoire from early pre-treatment Lyme disease. So this is acute, untreated presentation, where there's many small clonal families indicated by the red arrows. So it's a very diverse response against multiple different targets. In contrast, when we look at PTLDS patients, certain patients don't have activated B cells. There's probably multiple different causes for post-treatment Lyme, and perhaps an aberrant immune response is a potential cause in a subset of those individuals. And so here's examples of two individuals who had elevated activated B cells, or a certain level of them in post-treatment, in their post-treatment Lyme disease syndrome condition. And you can see in general there's larger families and there are fewer. There's, a certain set of families are expanded. And so further, we see evidence of families that have both shared IgA and IgG members. So there's an example on the top right where there's both. It suggests that the mucosal or the skin or the surface of the body initiates the response, and then it class switches to the more conventional IgG form of immune response. And then we also have ones that are either solely IgA or solely IgG. And then we're taking our antibodies and we're using them to identify the targets. And this just shows a scheme of arrays and immunoblotting that we use to identify the targets. And this is focused Lyme arrays where we take the antibodies that are expressed, and on the left side we have individual antibodies that bind individual Lyme proteins such as OSP-A, decorin binding protein, OSP-C. And on the right, different sets of antibodies from these activated B cells. Some of them actually bind host antigens. And so when we annotate the repertoires, so example, this is a repertoire from a patient with late disease, and you can see that certain families are targeting Lyme proteins in Borrelia burgdorferi, such as OSP-C, VLSC, decorin binding protein, and others are targeting certain human antigens like histones and fibrinogen and vimentin, which are known autoantigens in other connective tissue diseases. So perhaps there's an element of autoimmunity in certain people with post-treatment Lyme. And now we're expressing much larger sets of antibodies from patients who cleared or patients who developed post-treatment Lyme, better trying to understand what the exact targets are. So I want to share with you one last piece of data, and that's a study that we did in collaboration with Monica Embers as well as Mickey Tall at Stanford, where we're taking these antibodies that we make that bind Borrelia, and we're determining their ability to kill Borrelia in vitro. And so the second to last bar on the right is the negative control. So this is full growth of the Borrelia. The blue bar is 24 hours, the green is 48. And if you go to the left, we have several of our antibodies against VLSC, against BMPA, against decorin binding protein that very potently kill and prevent Borrelia growth. And now we're expanding this screen, trying to identify antibodies that might have therapeutic utility, likely in combination with antibiotics. So in summary, sequencing antibody repertoires in acute Borrelia-infected patients revealed increased activated B cells or plasmablast in people who return to health, that there's differences in repertoire isotype usage as well as clonality in people with different stages of disease, that in people with late-stage disease, they have antibodies that bind both Borrelia as well as to host antigens, suggesting an ongoing immune response against both. And we've identified recombinant antibodies that potently kill Borrelia. And so we're now working to kind of further screen and identify the most effective antibodies and to do what we call weaponizing them, meaning arm them to make them even more potent killers of Borrelia. So just in summary, we're taking samples from high-value patients. We're sequencing their antibody repertoires, specifically focused on these activated B cells. We then use this information to informatically identify antibodies of interest. We express them, characterize them, and we're hoping to use and leverage these to develop next-generation diagnostics for Lyme disease, as well as potential next-generation therapeutics to kill and control Borrelia infection, and to promote clearance of, for example, reservoirs. Just recognizing the individuals in my lab, Nitya, Shani, Lisa, Radhika, all drove these studies. We collaborate with many individuals listed on the right side. Thank you very much.